Hi. Uh, Now we're going to talk about the regulation of the pentose phosphate pathway. I mean, I really like this pathway because, as you can see, it's extremely flexible, and you can adjust the output exactly how you want it, how the cell wants it. And so let's see how this happens. By the way, most of the regulation happens, in people, it happens in the liver and the fat tissue. And the very first reaction, of course, that takes in the oxidative branch that takes glucose 6 phosphate and gluconolactone is regulated by NADP plus and NADPH. The more NADPH, the more product you have NADPH, the more it acts to inhibit the enzyme. However, if you have a lot of NADP plus, so non-reduced oxidized NADP in the cell, then you activate the enzyme, which kind of makes sense. This is a scheme that I like to use to describe the pentose phosphate pathway. So it's these parts here. We have the reductive and the oxidative parts. So we start with glucose 6 phosphate. That's the key one. That's the entry. This is where the carbon comes in the form of glucose. From the oxidative part, you get a 5 carbon unit, which can be, it is ribulose 5 phosphate, and then you can convert it right away to ribulose 5 phosphate and use it for amino acids, for a little bit for amino acids, but for nucleic acids. And of course, a major product is reducing power in the form of NADPH. Now, these 5 carbon units, we can interconvert them in a fully, it goes backwards. It is a fully reversible part where the 5 carbon units from the pentose phosphate pathway can feed into glycolysis and vice versa. Okay, so that's important. So, why would you want to feed the 5 carbon units from a pentose phosphate pathway into glycolysis? If you don't need them for anything else, you might as well just put them into glycolysis, maybe use some intermediates there, or just burn them all the way down to to CO2 and get some energy through the TCA cycle and the oxidative phosphorylation. So nothing goes wasted. Um, let's say, however, that all you need is the 5 carbon unit and an ADPase. So all you need is really to run the oxidative part, which is fine. And again, why would you need that? Maybe you are a cancer cell and you want to grow, so you need a lot of NADPH to do your biosynthesis, including, let's say, new membranes, which consume a lot. And you need to make a lot of nucleic acids, like new DNA, to synthesize. So that's all you need, and that's where the pentose phosphate pathway stops, just at the oxidative branch, nothing else. So you eat glucose that comes in from from the food, and you just generate these two key products that you want from the oxidative part of the pentose phosphate pathway. So that's fine. That's one way. Then let's say that all you need is just the ribose 5 phosphate for whatever. Let's say just to make DNA, nothing else. Well, then you don't do the oxidative part. You just funnel through glycolysis. You know, you run everything we talked about in the reductive part of the pentose phosphate pathway, you run them in reverse. Instead of generating glycolytic intermediates, you take the glycolytic intermediates, fructose 6 phosphate and glyceraldehyde 3 phosphate, and convert them exactly as we described, but going backwards now, to ribose 5 phosphate. Right? So that's fine too. So you can shut down one or the other and get the products you want. Um, or you may just need NADPH. Then you don't shut down anything. Okay, both go, both the reductive and the oxidative part. Why is that? Because from the oxidative part, you get your NADPH. That's the reducing power you need. And then you have no use for the carbons, really, the 5 carbons here. And you just take them all through back into glycolysis and then let glycolysis decide what it's going to do with it. So maybe burn them all the way down to CO2, as I said. So in that case, both the reductive and the oxidative part is on. So extremely versatile, very important pathway. In terms of human disease and its involvement, it turns out that the first enzyme we saw, glucose 6-phosphate dehydrogenase, mutations in that enzyme is by far the most common metabolic disorder in people. Okay, there are, you know, at least 500 million people walking around with a glucose 6-phosphate dehydrogenase deficiency. It is X-linked. Okay, and why is it maintained in our line here in our species, even though we'll see it can cause some problems? Well, because it can protect us from other bigger problems. Okay, such as malaria, which is endemic in many parts of the world still to this day. And it turns out that people who have this mutation and the glucose 6-phosphate dehydrogenase deficiency, they are protected from malaria. So, however, for the rest, there are side effects, all right? So there are problems. I mean, you don't just, yes, you may be protected from malaria, but actually you are prone to anemia. I mean, it causes anemia. Why does it cause anemia? Because in erythrocytes, in red blood cells, the pentose phosphate pathway is the only source of reduced glutathione. So we have not talked about glutathione. We're going to see glutathione a little later when we talk about amino acid biosynthesis. But it's a very small tripeptide. It's got three amino acids only. And it can be in the oxidized or the reduced form. It's got the sulfhydryl groups. And if it's oxidized, it makes a disulfide bond. And it is this reducing power on the sulfhydryl groups that it is used by all cells, use glutathione. If there is some peroxide, or it can actually detoxify oxidative stress, essentially. So if the cells are under oxidative stress, glutathione is summoned and does that. But of course, to regenerate glutathione in the reducing form, okay, every time glutathione goes and rescues us, it gets oxidized. And to get back into the reduced form, the GSH that you see here, you need an ADPase. You need reducing power. And who provides it? In this case, in the red blood cells especially, is the pentose phosphate pathway. So without the pentose phosphate pathway, red blood cells, they cannot deal with oxidative stress, and they lyse, and they die, and you get anemia. Okay, so that explains the hemolytic anemia if you have a glucose 6-phosphate dehydrogenase deficiency. So, but then again, that's a cost. But if you face this versus, hemolytic anemia due to oxidative stress versus malaria, on a population-wide base, that's how evolution works, maybe it's not so bad to have a pentose phosphate dehydrogenase activity, because that protects you from malaria. How does it protect you? Through exactly the same mechanism, actually. The malaria parasite gets into your red blood cells. It needs them to be alive, okay? But as it gets into the red blood cells, it causes oxidative stress. So a red blood cell that has defects in the pentose phosphate pathway will lyse when it gets infected by the parasite, and the parasite will die too. So that's the protective aspect of it. Which explains why it has been maintained in the human lineage to such high degree. As I said, 500 million people are carriers for the dehydrogenase deficiency. Now, it turns out that any oxidant, okay, will cause anemia in these patients. Any oxidative stress, it could be the malaria parasite, or it could be something else. And fava beans, as it turns out, will cause anemia in this patient. So they shouldn't be eating fava beans, and because they are very rich in oxidants. And this goes way back to Pythagoras of Samos, who, of course, we know the Pythagorean theorem. You've all heard it. It's kind of a cute story, but he was also kind of, in addition to being a mathematician, he was also a cult leader of sorts, and he was pronouncing all these edicts for his followers, and one of which is don't eat fava beans, right? So, and I don't know if obviously he didn't know the molecular mechanism of this, but I'm sure people must have correlated the sensitivity to fava beans to some subjects, some fraction of the population. The legend has it that his enemies, when they chased him off some city, Greek city in Sicily, he had to walk through a fava bean, field, and then he died. Okay, from acute oxidative stress, I guess. So this, we're not going to do it, but when the class was not online, I mean, we closed the pentose phosphate pathway with a sing-along from the discoverer of the pentose phosphate pathway, Horeker. And he had this mnemonic that you can read aloud and sing along, and that helps you memorize the whole pathway. But we're going to stop at this point, and I'll see you at the next video.